So basically, so after I graduate, start with the postdoc first, and then after my postdoc fellow, I become a research fellow. So right now, I'm a research fellow.
Okay. Today, Dr. Pipat will talk in the topic of human intestinal microbiota, the hidden gem in the gut. I will share screen for your slide.
All right. So
okay.
Do you give me the? I cannot click it. Wait. Did you see it? Wait. Is that okay for this slide?
Okay.
All right. So good morning, everyone. So yeah, I'm Piewngam. So today I will talk about the human intestinal microbiota. So the hidden gem in the gut. So what we will cover for today. So I will start with the human microbiome 101. So basically we will talk about what is the microbiome. And then I will talk a little bit in the trend in microbiome study. And then I will move to microbiome in health and disease. And later on, I will talk about the future trend. Oops. Okay, so, and then I will talk about the future trend in the microbiome study. And I will close with the current problem and controversial in this study. So let's start first. What's microbiota? So basically, microbiota defines all microorganisms that including a variety of bacteria, fungi, archaea, protists, and viruses, found in defined environment of the human body. For example, if we're talking about the intestinal microbiota, meaning that they all microorganisms that live in your intestinal tract, this is microbiota. And then they have another word. So what is a microbiome? Basically microbiome is referred to, a similar way to the microbial organism that reside in an environmental niche. And strictly speaking, this term describes their collective genomes. For example, again, if you're talking about the bacteria or the viruses that live in the intestinal tract, and then you collect their genomes, so we call intestinal microbiome. So this is the difference between microbiome and microbiota. So when this thing start, so basically the first bacteria was isolated, around 1880. So the first bacteria in the gut is Escherichia coli, and later on they have, Robert Hankett developed the technique that can isolate the anaerobic bacteria. Basically, in the gut they have mixed between aerobic and anaerobic bacteria. So Dr. Hankett developed a technique they call the Hankett technique. So this thing you can isolate the anaerobic bacteria. So now we found more bacteria in the gut. And the later on, you have high technology. And then in 2000, around 2000, they have the sequencing method was introduced. And then people can get more and more bacterial species. So, and then when the microbiome really start, so in about 2007, the NIH or National Institute of Health launched a project they call Human Microbiome Project. And 4 years later, they have two big publications in Nature. And these two publications, people consider as the first microbiome catalog So basically, if you study microbiome, you have to cite these two papers because they're talking about the structure, function, diversity of the human microbiome. All right. So when we're talking about the microbiome in the intestinal tract or GI tract, they define as a three-part. So the first part is from small intestine, and the second part is colon, and the last part is like the descending colon or close to rectum. By this picture, you will see that they have difference in terms of bacterial load. You will find more bacteria in the colon when compared to the small intestine. Because of what? Firstly, because of pH difference, because of antimicrobial peptide difference, and also because of oxygen difference. So in the colon, you have less oxygen, and then you have more anaerobic bacteria. So this picture is like a big concept, in terms of intestinal microbiota. And when you read a paper about the intestinal microbiota, you will see two big phylums. The first one is Bacteroides or Bacteroidetes, and the second one is Firmicutes. So people are talking about, Bacteroides and Firmicutes a lot because this is two big phyla that we found in the gut. And again, so as I introduced earlier, in our gut, they don't only have the bacteria, so they're composed of fungi and viruses or the archaea. So right now, they have a lot of study on bacteria, fungi, and viruses. And this figure show, some part of our GI tract start with the esophagus. You can already see the bacteria and some fungi in the esophagus. And again, so in the stomach, you have low pH. So basically some bacteria or some fungi can survive in the stomach. So, and later on, like they have small intestine. So for the small intestine, you have three parts. You have duodenum, jejunum, and ileums. So these things you will see difference in terms of bacteria and fungi. And the last part is colon. So colon is the big part that you found a lot of bacteria, fungi, and viruses. So this is the basic concepts that you have a lot of varieties of microorganisms in your GI tract. And how these bacteria or how these microorganisms provide you, the health or provide the benefit for your health. So we call colonization resistance. Basically, the bacteria itself or the fungi itself can produce some peptides that kill the pathogen, for example, C. difficile. So some good bacteria, for example, Bifidobacterium species, they produce the bacteriocin. The bacteriocin is antimicrobial peptides that can kill the C. difficile. This we call direct colonization resistance. And again, they have indirect colonization resistance. For example, once you have the good bacteria contact to the gut epithelial cell, these bacteria can activate the immune response. For example, they can activate the T helper cell, specifically 17 cell. And these things, after they were activated, they can induce the secretion of interleukin 22 or interleukin 17. And this interleukin, can activate the gut immunity, and then the gut immunity itself produce antimicrobial peptides and also kill the other pathogen, for example, E. coli or Cetobacter rodentium. So this is why you have to have good bacteria in your gut. When we're talking about the gut microbiota, so people we're talking about like, if you have shift in terms of the gut microbiota that can harm yourself, or sometimes you have to keep it balanced. But by this figure, within 24 hours, you will see the temporal modulation of the gut microbiota. But they have the main three parts in your life. For example, after when you are a baby, you have a big shift, the first big shift in terms of the gut microbiota modulation. And when you were adult, this thing will change it again. And then when you become the elderly individuals, the gut microbiota will change it again. But when they change it, they keep balance in terms of good bacteria and bad bacteria. That's why you still, healthy. But whenever this thing is imbalanced, you will have a problem. So I will talk this later. So the intestinal colonization resistance is the thing that provide the benefit for your health. But whenever if this bacteria is imbalanced, you will have a problem. And those things we call intestinal dysbiosis. The intestinal dysbiosis refer to the imbalance in terms of the taxonomical composition or the metagenomic function of the microbial community. For example, they have two main things of the intestinal dysbiosis. The first one, bloom of pathobionts. The pathobionts mean bad bacteria or bad microorganism. For example, the bloom of the Enterobacteriaceae in the gut in the people with the IBD disease. And the second one, the second part of intestinal dysbiosis is loss of commensal. The commensal refer to good bacteria. So basically you have less good bacteria in your gut. For example, if you lost some Bacteroides facialis, and they said that they can impact the disease of severity. For example, the development of the bowel disease, for example. How can you keep the bacteria balance, or what things can cause the imbalance in terms of the gut microbiota? The first one, infection or inflammation. The second one, I think this is the big one, is the diet. So diet itself can cause imbalance of microbiota. The third one is synobiotics, all the things that you take it. For example, the drug, whatever the chemical things that you take almost every day. These things also can cause the imbalance of microbiota. The fourth, hygiene. If you wash your hand all the time, sometimes those things can cause the dysbiosis, like skin microbiota dysbiosis. And the last one is genetic itself can also, cause intestinal dysbiosis. You start with the healthy microbiota, and these 5 things can cause dysbiosis. And once you have the dysbiosis, you develop the health problem. For example, your brain, sometimes they have like the gut-brain axis. If you have imbalance in your gut microbiota, those things can cause the brain problem. For example, stress, autism, or multiple sclerosis. And also these things can cause the systemic diseases, from the brain to the tools, to the food, for example. And the systemic diseases that I'm talking about is, for example, the type 1 diabetes, or atherosclerosis, or rheumatoid arthritis, for example. So what is the mechanism that link the gut microbiota and synobiotic metabolism? I'm talking about the drug, for example. So the drug, some drug, you start with the product, and then these gut bacteria can metabolize the product, and this product become active metabolite. And also some active metabolite, if they meet some gut bacteria can further metabolize this active metabolite, and this thing become inactive metabolite. So you get less active metabolite. Or the drug itself can kill bacteria, that toxic metabolite. For example, if you have 2 patients and one respond to the drug and the second one didn't respond to the drug that much, so this thing can happen. Maybe depends on the gut microbiota, because, for example, like I told you, the gut microbiota itself can metabolize those active metabolite or active drug, and become those active metabolite become inactive metabolite. So you get less substance to treat that diseases. So that's why some patients respond to the drug, but some's not. All right. So now I will talking about the link between the intestinal dysbiosis and some health problem. I will start with the neurological disorder. So when we're talking about the neurological disorder, we're talking about the gut-brain axis that I told you. So the first one, the major depressive disorder, or MDD. So they found that these patients, they have less commensal, remember the intestinal dysbiosis, they have two main mechanisms, the bloom of proteobiomes and loss of commensal. And major depressive disorder patients, they have both mechanisms. So loss of commensal, for example, probiotic or coprococcus, for example. And also they have bloom of proteobiomes, for example, flavonifactors or Bacteroides enterotype 2. And the second disease, Parkinson's diseases. Some patients respond to the drug, the dopamine. We use dopamine to treat the Parkinson's disease, right? Some patients respond to the dopamine, but some is not respond to the dopamine. And then they found later that people who has a lot of enterophyclis in the gut, those patients become less respond to dopamine. I will talk later that why. And the last example, the autism spectrum disorder. And they found that if you have a lot of intestinal candida, this thing can drive the pathogenesis of autism spectrum disorder. Okay, let's start with the dopamine metabolism. Remember, I told you that some patients, for the Parkinson's disease patients, some is not respond to dopamine treatment because they have the E-Fecalis in the gut. And they found that the E-Fecalis can metabolize the levodopa, by produce some enzyme, and those become a dopamine. And then this dopamine can be metabolized further by the Escherichia lenta bacteria. And after they metabolize, they become M-tyramine. And this M-tyramine cannot pass to the blood-brain barrier. So you have less L-dopa in your brain. That's why those patients is not respond to the levodopa treatment. This is just an example that somehow, your gut bacteria have the link to some disease, even some disease in the brain. All right, so we move further to the GI tract disorder. For example, the inflammatory bowel disease or IBD patients. This group of patients, they have a lot of proteobiomes. For example, invasive E. coli or IX, or they have a lot of Bacteroides fetilis. And again, so another disease is irritable bowel syndrome. It's like similar kind of inflammatory bowel disease. So these patients, they have a lot of bad bacteria. For example, Ruminococcus or Clostridium Clostridium species, for example. And the last one, the alcoholic hepatitis. So the alcoholic user disorder, some can develop to the hepatitis, but some didn't develop to the hepatitis. Why this happened? And they found later that patients who can develop to the hepatitis, they have a lot of E. fecalis in the gut. Again, the E. fecalis play an important role in the, a lot of diseases. Oops. What happened? Did someone click my slide? Oh, it's frozen. I cannot share my slide. Internet, can you help me?
I will try. Thatมันเปิดไม่ได้เนี่ยมาดูกดนะ Okay, Dr. Pipat, I will close
Yes.
and open again. Okay?
Okay.
Okay.
Okay. Okay.
Okay.
Okay. All right.
Okay.
Thank you. Where I will go very quick. All right. Oops. Okay. I think this slide. Okay. Yeah. So you know about the alcoholic hepatitis. Some patients can develop the hepatitis because they have a lot of efficacy in the gut. So why this happen? Because the efficacy cell, you know, produce a lot of the cytolysine that is toxic, like enterotoxin in the gut, and this cytolysine can destroy the gut epithelial cell, and those this cytolysine can destroy the liver tissue, and those patients become hepatitis. This is just an example of the intestinal dysbiosis to the GI disorder. All right, and then I think the last one is intestinal microbiota in infectious diseases. So when we're talking about the intestinal dysbiosis in infectious diseases, so we like always talking about the C. difficile infection, and also again, so enterococcal sepsis. Let's start with C. difficile infection. The top panel is like in general, like you have a lot of good bacteria. These good bacteria can produce or can activate the gut that produce the IgA, and this IgA can also inhibit the C. difficile. And sometimes, you know, the special characters of C. difficile is they can become a spore. And whenever, you know, you have a lot of good bacteria in the gut, C. difficile cannot germinate because, you know, they have the balance of good bacteria. But whenever you have less good bacteria, and this C. difficile can germinate and become a vegetative cell. And after C. difficile was germinated, they can destroy your gut and then cause the colitis. And this is the big problem right now because you cannot treat them with antibiotic. This because first C. difficile become resistant to a lot of antibiotics, and this body is and antibiotic cells can kill the other bacteria, can kill the good bacteria, and after they treat with antibiotic, the good bacteria was depleted, and C. difficile become really aggressive and cause colitis in the patients. So that's why right now the doctors try to find the new way or the new approaches to treat C. difficile infection instead of treat them with the antibiotic. The first approach is fecal microbiota transplantation, or FMT. Basically, like I said, you know, you have less good bacteria and C. difficile become aggressive. And doctor think about that, okay, why don't we just introduce the good bacteria to the gut, you know, and then, you know, C. difficile become less aggressive. By this thing, they transplant the feces. They have to form of the fecal microbiota transplantations. First, you eat the feces tablets or feces pill, basically you take the drug, or the second thing is that you inject the feces solution, to the rectum. This thing is fecal microbial transplantation. So they have a success of this treatment. So some patients received the fecal microbiota transplantation. They have best, C. difficile, have less C. difficile in the gut, and they become healthier. But later on, this technique has some limitation. They found that some patients get fecal microbiota transplantation die after, because those feces that they get have some resistant bacteria, and they call the bacteremia. So, and then doctor try to move to another approach, because this thing, it doesn't work because they still have some resistant bacteria. So first, you have to have the best screening before you do the fecal microbiota transplant, or you try to find another approach. The study from Thailand, we found that even you are healthy, and then you have a lot of these bacteria in the gut, 54% of healthy individuals have ESBLE, the extended spectrum beta-lactamase enterobacteriaceae in the gut. So this bacteria, this enterobacteriaceae resistant to ESBL drug, even you are healthy, you have this thing in your gut already. This is really a big problem right now. So because those microbiota transplantation have some limitation, the doctor or the scientist try to move to another choice. And what is the alternative choice? Obviously, the first one is probiotic. So the probiotic is the live microorganism that, when you administer in adequate amounts, confer a health benefit on the host. So basically, you take the live bacteria, and these live bacteria become, provide the health benefit to you. And this thing already successful for the restore the normal microbiota transplantation and function in antibiotic treated and C-section born infants, and now they have a lot of these things study a lot. For example, they also have one study, they use probiotic to treat the obesity. They use the probiotic as a supplementation for the overweight patients or obesity patients. And then these patients become they lost some weight, and the diabetes become better. And the last one, for example, we also use probiotic for to treat bacterial colonization or infection. And this study found that when you use the bacillus species, is that the probiotic, you can treat or you can interfere the Staph aureus intestinal colonization. This is the advantages of the probiotic. Okay, and I think this is the last one. So I just talk about a little bit about COVID-19. Because COVID-19, they have the receptor, the virus bind to the ACE2 receptor, and in your gut, they also have ACE2 receptor. So whenever this virus bind to the ACE2 receptor, they can, interfere the balance of the gut microbiota. That's why some patients have diarrhea, and and after they get a COVID-19 infection, they have like a diarrhea or they have a GI problem. And they have one study found that if you use, the consortium of the probiotic, that patients can have less diarrhea, and the lives become better. And right now we only have the top, the top approach of the probiotics. Basically, you get the bacteria, you give to the patients like randomly, but a lot of scientists thinking about the precision probiotics. Basically, the probiotics just for you. For example, the patients A, only get the probiotic A. The patients B only get the probiotic B. This thing can, directly treat for some diseases. We call the precision probiotics or personalized probiotics. All right, so right now we are at the beginning to exploit to achieve the precision and editing of the microbiota composition. As I show you, you only know that what is increased, some bacteria increase, some bacteria decrease, but we don't know really what is the real mechanism that, involved in some diseases. And they're frozen again. Anyway, I think that is my last slide. So, okay, maybe you can see this one, Okay, so when we're talking about the probiotics, so they become like increased interest in the use of probiotics, but the assessment, the risk and the benefits is urgently needed. Especially, you have to provide more evidence from the animal study and also from the human study to, provide the potential of biotherapeutic approaches. And this is also, I will take the questions.
Okay, anyone have question for Dr. Pipat? Dr. Pipat. Hello.
Yes.
Dr. Pipat, I mentioned about the imbalance of the microbiota in our gut. What is the most reason that cause for imbalance in the bacteria in our gut?
Yeah, so I think the main factors that can cause imbalance in the gut microbiota is diet, because you eat it every day. For example, they have some studies show that if you eat the red meat, for example, beef or pork, like a lot, almost every day, those things, they can, after you eat the meat, right? So you have some metabolize or metabolism in the gut, and those things can kill some bacteria, and you become the imbalance in terms of the gut microbiota. So I think the main factor is diet.
The compare between if I too many wash the hand and I receive the drug resistant for the bacteria. What the reason is the more causing to imbalance of bacteria, but too many wash my hand and take for the drug.
Right. Yeah, so those things can also like the main problem. So that's why a lot of patients, but they have a lot of patients when they were treated with some antibiotic, they become a more problem after the antibiotic treatment. And for the hygienes, they have the terms of the hygienes theory, right? So they said that, oh, if you, for example, you have some vegetable, you buy some vegetable from the market, and you really hygiene, so you wash them a lot. So basically, you just wash out all the good bacteria, and you didn't get any bacteria from that veggie. And again, so for the hand washing, they don't have study yet, but like for example, the study from USA, they said that you should not take a shower too much, because after you take a shower, so you deplete all the skin microbiota. That for example.
Okay. Any question from the room? Okay. Thank you so much, Dr. Pipat,